So we start with this self-review on histology. And everybody should see the picture. And it's different cells at different stages of mitosis. If you pick a cell, it'll line up, and this will say interphase. I know it's kind of blue right here, but interphase is where the cell is all just nothing's lined up, nothing's organized yet. The chromosomes, the chromatin is all organized for the cell, and the cell's working. So that's your interphase. Okay. So again, if we go back, where was our interphase? Okay. So this one here is interphase. It's set up a little bit different. I'm just trying to get the blue to go away. So here's your interphase. You can see right there, everything's just all inside the cell. The cell's just working. Nothing's organized here. Remember, what are the 4 steps of mitosis? Prophase, metaphase, anaphase, telophase. Okay. Prophase is where the chromosomes condense. Metaphase is where they line up in the center. Anaphase is when they start to pull apart. Telophase is where the cell actually starts to have cleavage and pulls apart. Okay? Those are your 4 steps. Okay. So if you look at the different ones, we have interphase here. And I think we can show labels. There we go. That's what I want to show. Okay, so this is your interphase right there. You can see the blue there. Okay, you can see where it's just lining up. Everything's kind of, I'm trying to get rid of the blue stuff. Everything's kind of just in the middle. Nothing's organized yet. Okay, prophase. Everybody see prophase in the bottom left corner here. Prophase is where you can see the chromosomes have started to form, right? Everybody see the difference between interphase and prophase. Look at interphase. See how it's all black and kind of spread all out? Okay. You look at prophase. Prophase, you actually see the chromosomes starting to form. Okay, you see the rods of the chromosomes starting to form. Then you go to metaphase. Metaphase is on the left-hand side here. Okay, it's where they all start to line up. You start to line up in the center. You see all the chromosomes now, instead of being spread out through the cell, once you're condensed, are now starting to line up in the middle. That's metaphase. Okay. Anaphase is where they start to pull apart. You see right over here on the, right below the mouse cursor. I can't write on the screens on these things anymore. Wish I could. Anaphase is where the chromosomes actually pulled apart. And then telophase is this one right here on the top right-hand corner. And you see where the 2 cells have separated and pulled apart each other. They have 2 new daughter cells now. So interphase is when the cells are just working. Prophase is when the chromosomes start to condense around the histones and coil. Metaphase is when they line up in the center. Anaphase is when they start to pull the chromosomes apart. And telophase is where the cells actually start to split and actually form 2 cells. So far, so good? Okay, there's 13 of these slides. We'll go through them all like that. Okay, and again, this isn't how you, this is what you'd see in lab under a microscope. All right, you see the different phases. Okay, remember telophase. Telophase is we have 2 cells. You see the 2 nuclei pulling together. Metaphase is where they're starting to line up. Interphase.
What?
There's no. What's that?
I'm sorry.
Oh, that was in the question. No problem. Okay. Interphase is where the cell, there's no chromosomes formed. It's called chromatin. It's all separated. They're not wound around the histones anymore. And then anaphase. Anaphase is where they pull apart. So if I show you these slides, you should identify the different phases the cells are in. We got another onion root tip slide. Okay. Can anybody see one that is in, oh, there's interphase right there. It says interphase right there. Okay. You can look at other ones and see interphase. If everybody see the pointer? If I point to this cell right here, what cell? What is that in? What state?
Interface?
Interface. Perfect. Because nothing's condensed. The DNA is in chromatin. It's all spread out. And here's a picture of your interface right there. Okay, right on this. You can see if there's nothing condensed yet, just starting to. Prophase. I don't like the blue around it. Prophase here on the left. You can see the chromosomes starting to form. The DNA starting to wrap around the histones and starting to coil and condense into chromosomes. And you start to see that. That's your prophase. Metaphase is when they line up into the center. This blue one right here. You can see how all the chromosomes are starting to line up. Okay. And right into the center so they can be split apart. And anaphase, you can see what actually starting to split apart right there. And does anybody see a telophase? This could be a telophase right here because it looks like these two have just formed and there's no cell wall dividing these. So this could be a telophase right here. Because again, there's no cell wall separating them. So they're two that have pulled apart. Okay. Again, more slides to look at through your microscope.
I had a question about the last slide. Why was there a blank line between where there was
a blank line where?
In the middle.
On the last slide?
Yeah.
Let me go back. A blank line.
Yeah, it looked only like there were cell walls, but nothing.
Oh, right up in here, yeah. Remember, this is a three-dimensional slice. So if you slice it where one of the layers doesn't have the cells, if an onion skin's round, it's only going to slice one of those layers like that.
Oh, okay.
You can see one of them, the line, like the line above it, is kind of not real dark. It gets more darker as you go. It all depends on how you cut the line through it. Does that make sense?
Yeah, thank you.
Yeah, you're doing your section, and some of the sections aren't going to show the whole cell. You got to remember, it's a three-dimensional object. You're putting on a two-dimensional field. Because you can see how light these are here, and how dark these are above it, and how dark these are, how light these are here. It all depends where the nucleus is, where you do your slice.
So the one that's
enough to get
three down, the one that's three down from what's labeled as metaphase is another metaphase, correct?
This one right here?
Yeah.
Yes, line up in the center. Perfect. That's another metaphase. Most of you are in interphase right here. Let me see something else. This is probably metaphase right here too. You see how they're starting to line up in the center. And this one here is prophase also. You see where they're starting to conglomerate around. So this would be prophase, that'd be metaphase. And like I said, this would be metaphase. This is actually a good example of metaphase right there. Nice deep color. Okay, that answer it? Okay. And don't be afraid to ask questions during lab. That's what these are for. These are for interactions. And again, onion cell, that's a perfect example of telophase. You can still see on this one right here, you can still see all the fibers that are connecting it as the centrioles pull the DNA apart on the opposite sides of the cell. So that's a good example of telophase. This is a good example of prophase. That's a good example of metaphase right there. I still come across it. Go from the top. There we go. Right there's a good example of metaphase lined up. Okay. Let me point to one here. Let's see. See this one? Well, that's still prophase.
Somebody just asked a question in the chat.
Oh, what's that? What are the light cells again? The light cells, okay. The light cells is where the section was cut through the onion and it didn't get the nucleus. So therefore they're lighter or just barely got the nucleus. That's why they're lighter. That's why they're not present. You got to remember, this is a three-dimensional object. Okay. And you're making a slice through it. So you're not going to hit the nucleus of each one of these. So some of these lighter ones, like right down here and all this, and some of these ones that are blank right here, those are layers where the nuclei is either above or below the slice that we made. Remember, making a real thin slice through this. That's what we're looking at under a microscope. Okay, so the darker ones would be good examples just because they're in the plane or the field that they're in the plane that we made the slice across. So you can see them real well. If they're below or below, you're going to get a little blurry vision. You're going to get some that are empty looking because the nucleus isn't in the area we made the slice. Does that make sense?
Yes.
Yep. All right, perfect. And let's see. See the cell right above my pointer here. What is that cell? Prophase, metaphase, anaphase, telophase.
It looks like a prophase going into metaphase.
I agree 100% with you. It's prophase. It's almost into metaphase. They're starting to line up, but they're not quite there yet. Okay, you can see metaphase like right here. I think those are all lined up. Everything's lined up in center. But this is their chromosomes. So there's prophase, and they're just starting to line up. So it's prophase just leading into metaphase. Perfect. Yeah, let's go to the next one. Takes a while to load sometimes from their website. You see all the different cells. Again, it's an onion sample, I think. Yeah. Onion root tip. All right. Perfect example of anaphase. Anaphase is starting to pull apart and separate. Not too many other examples. Most of these other cells are in interphase. Don't see too many good examples of anything else. But it's a really good example of anaphase. That's a test one right there. That's when I would put on a test and say, what is that? Because there's no doubt about what that is. Okay, it's past prophase, it's past metaphase enough, and the things are starting to separate, but they haven't started pulling apart all the way to the cell yet. The cell hasn't had any cleavage. Okay.
Anaphase.
I even told you it's anaphase. I click on it. All right, so now we've got metaphase. You can see them lining up. Interphase, where the chromosomes are not formed yet. They're still in chromatin phase. You can see anaphase, where you got the separation. And you can see telophase, where you can see right here. You can see the cell wall right in between them, just starting to form. I'll take that blue stuff off there. You can see the division just starting to form it above there. Okay, let's say where the pointer is. What's this one? Any takers? What's that right there?
Either a prophase or metaphase.
Perfect. Prophase. I'd say prophase because you still have these chromosomes that are still out here not lined up. Now here you see they're lined up. So this would be what?
That would be the metaphase.
That'd be metaphase. Perfect. Got it. So you can see metaphase, interphase, telophase, and anaphase on all those. Real good examples. And this is a whitefish cross section here. And this is a H&E stain. It's a little different stain. But on these ones, you can see the anaphase. I want these to line up here. Right. Okay, anaphase as they pull apart, both those two. You see that real good right above my cursor. I don't like how, because once it highlights, can you still see it on your screen?
No.
No, it blurs it on mine too. So if you look at this one here, this is anaphase. Let's go to another one. You can see right there where they're starting to pull apart. Telophase, you can see right here. You can see right here where the dotted lines come in. That's your cleavage. That's where the cells are starting to pull apart. You can see it if you take it off the cursor right there. Okay, you can see metaphase. All three of these are metaphase. Okay, look at this one right here where my pointer is. That's metaphase also. Excuse me, that's metaphase also. They're all lined up in the center. That one's metaphase. Okay, you can see someone just starting to pull apart. Okay, prophase. You can see just this thing just goes crazy when it goes across here. You can see this one is prophase right here. All starting to be condensed and just starting to line up. They haven't lined up yet. So this is your prophase. metaphase, telophase, and then anaphase as they start to pull apart. Trying to look for different examples. Okay. I guess a couple more of these slides, kind of getting repetitious. It's all the same stuff here. But you can see metaphase, they're lining up in line. Anaphase, you can see them just starting to pull apart. Prophase, you see everything starting to condense. And you can see telophase. And let's look at, is going to mark that one too as telophase. Yes, mark both those. Okay, telophase. Again, you see the invagination. You see the indentation here, the cleavage. So if you look at this one here, that's what's blue, and this one here, what's the difference? They're starting to pull apart here.
The cell wall.
Right. The cell wall's not invaginating. So they're starting to pull apart, so that's anaphase. Okay, so this is anaphase. Okay, the reason this is telophase is you have cleavage where the cell wall is starting to divide right above each of those. You can see it real well right here, just below the mouse. If I hit it there, it turns blue, but the cleavage is right there. So you can see the cleavage starting to form. Once you form that cleavage, then you've got anaphase. Telophase. Okay, so that's the big differences. Okay, metaphase, lining up, prophase, you're all coming together. I'd give you, if someone wanted to say this was metaphase, I think that's pretty close because it is starting to line up in the center, but still prophase going into metaphase. Anaphase pulling apart. There's no cleavage between the cell yet, so this is still anaphase, and then metaphase. And if I pick the cell here, like where my pointer is, what would you say this one is? Right where the pointer is, at the top of the screen. Metaphase?
Mm-hmm.
I would agree, yep. And this one here?
Also metaphase?
I would accept metaphase or early anaphase. Okay, you can see the spindles here.
Mm-hmm.
You can see them just starting to pull apart. So I would accept either one on this one. But this one definitely metaphase. This one metaphase or early anaphase. Perfect. That's a real good example of prophase right here. Prophase is just, it's not lined up yet, but the chromatin is starting to condense and form chromosomes. Anaphase, you see them pulling apart. Metaphase, see them lining up in the middle. I don't see anything else down here. What's this one right here?
Metaphase.
Metaphase, perfect. And this one here would be?
Anaphase. I mean prophase.
Perfect. No, I'd say anaphase because you see the fibers just starting to pull apart right here. You just can't see them here because they're three-dimensional. Not a great picture, but I'd say anaphase. I can see how you would say prophase though. But since I can see the spindles here pulling, I say just starting anaphase. All right. A couple more. Again, metaphase, anaphase, you can see them pulling apart. That's a real good anaphase right there, this top one right here. That's a real good picture of anaphase. And the rest are just interphase right there. Three more. And this is just prophase blown up a bit. I don't think it's the
Prophase.
computer intelligence prophase. Okay, all the chromosomes are starting to condense, but they're not lined up in the center yet. Okay, so that's your prophase. You can be on Pearson for this, or you just watch what I do. You can access this Pearson slides later to go through everything. It's no problem. You don't have to be on it right now.
Okay.
No problem. And last one. That is, what is that?
Anaphase.
Anaphase, being pulled apart. And all the other cells are in interphase. I don't see anything else even condensing. Okay. That was the cells on that. Let me run through the quiz quick here. Okay. let's see if we can do this, if this works out on our screen here. Where am I going to do this? Self-review. The highlighted cells. Okay, right where the pointer is here. These highlighted cells. These ain't turning on off the highlight. So look at the highlighted cells and tell me what phase they're in. What would you think? Those two.
Would it be interphase?
I would agree with you. I'd say interphase because there's nothing condensed. You're right. Number 2, a highlighted cell. Okay, which phase of the cell cycle? What is that on?
Phase.
With the pointer.
Anaphase.
Anaphase. Okay. And the highlighted cells in which phase of the cell cycle? Where's your oh, there's your highlighted cells. What does that look like on the bottom left?
On where?
Oh, there.
I'll turn on the highlight. There's your highlights. That one here and this one here.
Prophase.
That would be prophase. They're condensed, but they're not lining up yet. All right. Some more of these. Okay, look at the highlighted cells. Turn on my highlights. Those two right there.
The Zoom meetings, for lack of a better term, it's lagging. So your voice is like 5 seconds ahead, or at least on my end it is.
It's the same.
Oh, okay.
Yeah, same here. He's going faster than it's going.
It's going faster than the slides. Let me see if I can. We got a bunch of people online right now. Yeah, it's probably going to lag a little bit. If I talk slower, will that make it better? Would give it time to catch up?
Or just whenever you turn. It's mainly when you change a screen that it happens.
Okay.
So if you change a screen, like wait a little.
Yeah.
Okay. Got it. All right.
Thank
So
you.
that should, no problem at all. Thanks for letting me know. This is, these two slides are what?
Prophase?
Looks like prophase to me. Okay. What are those? Is that better now? I'm catching up.
Yeah.
Okay. What two slides are these?
Interface.
Interphase, right? Everything's spread out. Nothing's condensed yet. I'd say interphase 2. So far, we agreed with them. It's pretty amazing. All right. Let's see internal highlights. So it's this cell right here.
Telophase.
Telophase.
That'll be what?
Telophase.
Hard to tell, but I'd say telophase. I think I can see the cell membrane right here starting to divide. So I would say telophase also. Correct. Okay. There's a cell. What is that one in?
I would say that one's in telophase as well.
Yeah, I think that's comparing the two cells there. Yeah, those are the two cells. You can see definitely see the cell membrane here. See the two daughter cells. So I would say telophase again also. And the blowfish here. Okay, that's cell right there. What would that be in?
Anaphase.
I'd say
It
either
looks like
late anaphase.
telophase.
Yeah, anaphase or telophase. It's. So I can still see some of the spins, but I think I can see invagination here and here, and a little bit of line there. So I'd probably say telophase.
I have a question.
Yes.
The last slide that you showed, I'm just curious if cytokinesis would have been an option, would that have been correct as well? Because it kind of looked like cytokinesis.
If it had, it's early cytokinesis. If this line extended down to this membrane here, if you could see it being complete across the entire cell, then yes. But it looks like it still might be forming. But I wouldn't mark cytokinesis wrong on that one.
Okay, thank you.
Okay, but it's because you got to remember the section could be cut at an angle where it just doesn't show that division yet. So you could make an argument that is cytokinesis. Okay, but yeah, very good point. And this we said was telophase, right? Yeah, perfect. All right, this cell here. What would anybody think? What's that?
Metaphase.
Metaphase, right? Everything's lined up in the center. I would guess metaphase. And we might not agree with these guys all the time, but so far we've been doing pretty good. Whoops, I didn't turn on the highlight. I hit the wrong button. It gave you the answer. The answer is metaphase, right? You can see everything's lined up in the center. That was my fault. I hit the button too fast. Number 11. Here's your highlighted ones. What would those be?
Would that be prophase?
I agree 100%. Everything looks like everything's just starting to condense. That'd be prophase. This one would be am I still lagging?
Yeah.
Yeah, okay, well.
Telophase.
I don't know. I'd probably say anaphase with that one. Yes, I don't see a cell membrane forming, and I still see all the spindles. I'd probably say late anaphase, maybe early telophase. I would accept either answer, though, on something like that, because that's kind of ambiguous there. But it looks more like anaphase to me. But there is no anaphase, is there? I would have to say telophase then. Okay. But I'd say late anaphase, early telophase. Okay, if we're not up here, so we're now doing this. No, this is all for your benefit. You can always access this later on. Come back in just to restudy. This is not a quiz that we're going to be doing or regrade or anything like that. This is just for learning right now, going through. So don't worry about the question or anything like that. But this is if you go back and you can go back and practice for your practicum and things like that at the end of the year. But telophase. No problem. All right. Let the lag catch up. All three of those. Turn on my highlights.
Metaphase.
All right. Those are lined up in the center. You got it. Those have to be metaphase. And these are
Metaphase as well.
Metaphase.
Metaphase.
say metaphase, but I can see spindles starting to form just a little bit. So very early anaphase, but I'd go with metaphase if I was doing it. Yep. Good. Some can be ambiguous. There we go. Okay. Perfect. Metaphase, right? All lined up in the center. Just a couple more and then go to the next section.
Couple more, please.
Oh, shoot. I hit the wrong button again. I had to turn off the highlights. Everybody see why that's interphase?
Yeah.
Because there's no chromosomes formed. It's all chromatin. It's not condensing. And my lights are getting. Let me turn the lights on in here so you can see. Your light's turned on. It's on my cell phone here. There we go. All right. It was getting darker. I didn't notice it. And this one has no highlights and has no arrows. So we have to guess. Does anybody see any arrows or highlights?
Nope.
No.
That's because it's asking which phase of the cells are most in this image.
Oh, helps I read the question, right? Which phase of cells are most of the cells in this image? Most of these cells are in what?
Interphase.
I say most of the cells are in interphase. Yep. Got a couple in metaphase, got some in anaphase, but most of them are in interphase, all these right here. Yep. Very good. Learn how to read the question. During which phase of mitosis do centromeres first occur on the chromosomes? Okay, which phase of mitosis do the centromeres first occur on the chromosomes? Okay, they line up in the center. The centromeres attach, and that's what pulls the chromosomes apart is the centromeres. So they first attach in metaphase, and they start to pull, and that causes anaphase to occur. So it should be metaphase. Incorrect. Prophase. I would still say metaphase. First occur on the chromosomes.
But don't the centromeres come up during late prophase?
During late prophase to metaphase, yeah. So I would accept either prophase or metaphase.
You said you would accept either or?
Yeah. I accept prophase or metaphase on that one for that question. Because you don't see them in prophase. You know, they're starting to migrate there. And in the highlighted phase of mitosis, the genetic material is in the form of what? So here's your highlight. Okay. What is this genetic material in the form of? Chromosomes, chromatin, centrosomes, or centromere? There's your two highlights. What phase is that in? First of all, what phase are these two be in? This one here or this one here?
Prophase.
Prophase, because you can see them. So what is the DNA material? What's the DNA material?
Is it chromosomes?
What's that? Chromatin's where it's all spread out. This is chromatin. This is chromatin where it's an interphase. Once you're in prophase, the chromatin wraps around the histones and they start to condense and they form chromosomes. So once you're in prophase, once you enter mitosis, the DNA material is in chromosomes. So it should be that one. And one more. In the highlighted phase of mitosis, each chromosome is composed of, turn on highlights. Okay, so here's your highlighted cell. So you're in what phase?
Interphase.
Anaphase or late anaphase, early telophase, right in here. Okay, so it says in the highlighted phase of mitosis, each chromosome, so each individual chromosome is composed of what? A single chromatid, extended chromosome, extended chromatid, two chromatids connected at the centrosome, or sister chromatids?
Sister chromatids.
Each highlighted phase, each chromosome. They're sister chromatids, but I think they want a single chromatid because it says the highlighted phase each chromosome is composed of, because these are already separated. I could do either one, a single chromatid or a sister chromatid. Which one you guys want to try?
Sister.
Sister? Got it.
Sister.
No, single. It's worded funny. Like I said, I would have accepted a sister chromatids. It's worded as singular. The highlighted phase by each chromosome, not chromosomes. So if it's singular, it'd be a single chromatid. sister chromatids refer to the two that are pulled apart. Now they're sister chromatids. You got two of them. So since it didn't have a plural, I would have said single chromatid. But that's being really picky. I would never be that picky on a test.
It would be sister chromatids and telophase.
Right. They're sister chromatids if you're comparing the two. If you're comparing one, they're single chromatids. Okay? It's just semantics. All right. So we went through that. That's good. And let's go back to histology. An expert quiz without sending it in. And we talked about epithelial tissue, right? Talked about simple, stratified, squamous, cuboidal, columnar, and pseudotransition. So if we look at different section, okay, simple squamous and simple cuboidal, kidney, this is through the kidney, it's a section, and see the corpuscles right here. And this is simple, squamous, and simple cuboidal. If you look at these right here, let me blow it up. So you can see it here. Okay. So simple means what? How many layers?
One layer.
One layer of cells, right? Okay, so this is your single layer right across here. Okay, and squamous because the cells are what? What's the shape of a squamous cell?
Flat.
Thin and flat.
Thin and flat, they're squished. Okay, so you can see around this opening between this here, you can see one layer of cells. Let me get rid of the blue ring here somehow. There we go. So right around here, if we look, I'm trying to get away from the blue ring here. Okay, look at this ring right here. That's your basement membrane. This is simple cuboidal epithelium. So where the ring is here, I'm going to get rid of the ring here. Okay, so look above it. You see one layer of cells, and you see they're all cubes. So right here, this is simple cuboidal epithelium. So one layer, okay, in the shape of a cube, and that's your epithelium surrounding this open, this lumen right here. Okay, everybody see why it's simple cuboidal? It's one cell layer thick. Okay, you can see how those are cuboidal. Now look at the cells above it. See how these cells are kind of squished? Right there, right there. All the cells inside this thing are flat. All the cells in this one are cubes. Okay, so it's simple cuboidal. This is simple squamous. Does that make sense? You can see the cubes. Simple cuboidal. You see the cubes right here too. Okay, this is simple cuboidal. This is simple cuboidal. And this is simple squamous. You can see how they're squished flat. Okay, not quite as easy to see as mitosis ones. Again, this is an enlarged view again. Now look around the edge. See where my mouse is going around the edges here?
Mm-hmm.
See where the blue circle is? Okay, I'm going to take the blue circle away. And look where that is. I'm going to take the blue circle away. There was a nucleus around here somewhere. I'll get rid of the blue circle. Please get rid of the blue circle. Oh, show labels. There we go. That's what I wanted to show. Show labels. Okay. Now, this is a nucleus. All these are nucleus right here. And this, no, it still doesn't do it. Hide labels. Nope, that's not what I want. I'm trying to get so it doesn't show that blue stuff when I talk about it. I can't draw on these screens with this program. All right. So everybody sees the right here. This is your blue ring.
Mm-hmm.
Okay. See how the cells are all flat? This is the white area here. This is the lumen or the space. What's lining the space is your epithelium. Remember the epithelium lines openings, lines your externa, and lines all the openings in the body. This is the opening, and these cells are very flat and squished, and there's only one layer of them. Okay? You see the nuclei, how squished they are right around here. All these are squished. and it's only one layer, okay, which makes sense in the kidney. That's the filtration area. Okay, so this is simple squamous epithelium. Okay, this is a surrounding vein. You can see the white area is a blood vessel. Inside the blood vessel, you have red blood cells. Okay, this is what you see looking through a microscope. Surrounding the blood vessels are all this smooth muscle, okay, and this layer here of epithelium. This is simple squamous epithelium, because you can see how it's all flattened around the blood vessel here. And you can tell this is smooth muscle because why? Smooth muscle has no what?
Striation.
No striations. Perfect. The smooth muscle that controls your blood vessels, and that's involuntary control. So again, this is simple squamous epithelium surrounding a blood vessel or vein, a venule. You can see the nuclei. And then this is simple squamous epithelium. This is squished attachment. It's a lot harder to see on this slide than the other ones because you're looking at like a cross section of it. But simple means one cell. Squamous is squished, and epithelium. So it's just a thin layer. Okay, simple cuboidal epithelium. This area right in here.
Your screen's lagging again.
This is the lumen, the white area. Okay. Let me know when it catches up.
We're caught up.
Okay, perfect. Thank you. All right, this is simple cuboidal epithelium. Blow that up. Okay, so look at all the white sections. And surrounding each opening, you can see why is this not squamous like the other ones?
Because it's in a cube shape.
Perfect. Perfect. It's in a cube shape right here. You can see all these cells are a cube. They're not squished. This is simple cuboidal epithelium. You can see all the nuclei there. Okay. Okay. Again, you can see the lumen, which is the white area. You can see surrounding that opening, these are simple cuboidal epithelium. Simple means one layer. Cuboidal means shape of the cube. Epithelium means the lining of the lumen or the outside. Okay. That would be a good test slide right there because that's easy to see. That's something I can use for a test slide because you can see your opening right here. You can see your lumen. You can see what surrounds the lumen is only one layer of cells. You see it here. You can see it here where my mouse is. You just see one layer of cells and they're cuboidal shaped. This is a real good one here too. Everybody can see where the mouse is? Mm-hmm. Okay, this is the opening. This is the lumen. And around it is one layer of cuboidal cells. So simple cuboidal epithelium. Okay, again, perfect example. You can see the gland, blood vessels going through the gland where your opening is. You can see one layer, and you can see it in the shape of cubes. So simple cuboidal epithelium. Okay. We'll give it a second here. So you can see the duct, which is a white area. And you see it lined with simple cuboidal epithelium. Okay. Okay, now look at the difference. You can see the area right in here. Okay, let's see if I can blow this up.
You're lagging for me badly.
Okay. You caught back up yet?
Yes, thank you.
Okay. Oh, no problem. Thanks for letting me know. Not a problem at all. Okay. Actually, I might try doing this on an iPad, see if it doesn't lag so bad next time. This is our simple columnar epithelium. Why is it columnar as opposed to cuboidal?
Because they're long.
Right. Look at the cells. The cells are longer than they are wide. They're a columnar shape. And there's only one layer surrounding the lumen. So it's simple columnar epithelium. And this is through the duodenum, which is the area where you absorb most of the stuff, your nutrients from the stomach. That's why it's a simple layer. It's one single layer. Okay. So this, you see, this is simple. It's one layer, right? Okay, you see the basement membrane, the opening, the top white area is your lumen. Okay. But what's on top of the cells? You see the microvilli, right? That's the small intestine, help increase surface area, increase absorption in that area. But it's simple columnar epithelium. It's one layer. And again, if you look at this, this whole white area here is the lumen. Okay. And again, look at the cells surrounding it. It's one layer cells, and they're columnar in shape.
That
it's
goblet
simple.
exploding.
What's that?
I said, is the goblet cell exploding? Why is it looking like
Goblet cells excrete. Is this the stomach one? Yeah, small intestine. Goblet cells excrete hydrochloric acid. And you can see something being excreted up in these areas right in here, into the intestine. Those excrete different materials, help with digestion. And we'll get into that when we do the digestive tract. We'll get into goblet cells and what they excrete. But it looks like this could be an excretion here from it, or it could just be an image on that three-dimensional cut. Okay. Okay, pseudostratified columnar epithelium. Remember, pseudostratified means the nuclei don't line up. Okay, so you can see the nuclei are at different locations. Okay, so you can't really tell if it's single. It almost looks single, it almost looks stratified. So it's called pseudostratified, which means the nuclei aren't lining up straight. So pseudostratified, and they're columnar in shape, and it's epithelium. You can see the cilia on the top of the surface where the lumen is. Now, can you look at that and find simple cuboidal epithelium in that same sample?
right in the middle?
Right in the middle. You got it. Right here. See where the mouth points? This is simple cuboidal epithelium. That's what's surrounding this one right in here. So see how the nuclei are all in the center here. They all make a line. The nuclei up on the top here are all different places. So that's what's called pseudostratified, and it's columnar because they're taller than they are wide epithelium.
Since it has a cilia, wouldn't that make it ciliated?
It has, really can't tell if it's microvilli or cilia. Yeah, you could call it, it would be, the efficient would be ciliated, pseudostratified columnar epithelium. You're right, because there's cilia up on top of it. You can see they're a little bit longer than the microvilli. So if you got cilia right across here on the top, it's ciliated, pseudostratified columnar epithelium. Yep, exactly. Okay, this can be the same way. You can see real thick cilia here. This is even a better example of it. So it's cilia. You see the goblet cells inside. But everybody see the nuclei in different positions. I mean, you can see the nuclei up top. See all the little blue areas right there? Those are all nuclei. The nuclei are in some are the basement membrane, some are the apical portion. They're kind of spread out. That's why it's pseudostratified. Because it could be simple, but it could be stratified because the nuclei aren't lined up. You can't really tell. But they're long cells, so they're columnar. So this would be ciliated, like I said, it's because there's still on top. Ciliated, pseudostratified, columnar epithelium. All right, so again, pseudostratified, this is ciliated. Okay, because the nuclei are in all different positions and they're tall, they're columnar. So ciliated, pseudostratified, columnar epithelium. Attached to the basement membrane there. Stratified, squamous epithelium. Stratified means what?
Multiple layers.
2 or more layers, perfect. And squamous is all these squished flat cells. Let's blow it up here. So you can see this is the basement membrane here. Okay. You can see as you move up into this area, these cells get squished and flattened out. That's your epithelium. So this is a stratified squamous epithelium. You look at the apical portion, you see the cells are just really flat and squished down as it's moved upwards. Okay. So it's stratified, definitely more than one layer. Squamous, because all the cells are squished near the top there, and epithelium. Okay. Again, stratified, squamous epithelium. You look at the basement membrane, those are stem cells that are grouped into different cells, but look at the top layer near the lumen. See how all those cells are squished down together. Some are even missing their nuclei. Okay, and they're stratified because there's multiple layers. Squamous because the cells are squished or flat, and epithelium because there's lining, a lumen, or an opening. So again, stratified, squamous epithelium. Okay, same thing here. Okay, you got modular cells down at the basement membrane down in here. As they move up, the cells become squishier and squishier, and it's stratified because it's multiple layers. So it's an area where abrasion can occur or trauma, so it has to be reinforced. Okay? It's not simple because it has to hold up to trauma in the area. So it's stratified, squamous epithelium. Okay, again, everybody sees it gets more towards the apical portion away from the basement membrane, more towards the lumen. The lumen is very flat, squished cells. Stratified, multiple layers, squamous epithelium. Again, multiple layers, so stratified, squamous epithelium. Okay. Again, this is your lumen right here. This is white area. This is the opening. Okay. There's opening into the duct right here. Okay. This is your esophagus. So a lot of food goes down it. Food traumatizes it. You have to have cells to protect it. A thick layer. That's why it's stratified. Okay, a little different section what you're looking at. You can see the basement membrane. You can see the multiple layers, and on top you have all this dead layer up on here too. So what we'll learn later. Let me get that up there. Okay, you can see the squish cells, and then top of the squish cells, you see all this red stuff right here. Okay, that's keratinized. Okay, no nuclei. It's dead tissue. It's like your skin out there. So in actuality, this is keratinized. Keratinized, stratified squamous epithelium. Okay, so this is keratin right here on top. Again, stratified squamous epithelium. This is the epidermis. You can see your dermal papillae here, which we went to, basement membrane, and stratified squamous epithelium. And this is keratinized, like we said before. See this right here on top here? All this stuff, these fibers, this is keratinized tissue. There's no nuclei. There's nothing living up in here. It's keratinized tissue. So it's keratinized, stratified, squamous epithelium. Because the cells near the edge here, these are all squished. All right, let's go here. 27. Okay. See the opening? Look where the arrow's pointing here. Why is that stratified?
They're on top of each other.
Perfect. There's more than one layer. They're on top of each other. You can see at least 2 layers, sometimes 3. Okay? There's a minimum of 2 layers around that opening. So that's stratified. Simple is 1 layer. Stratified is 1, 2 or more. So that's stratified cuboidal epithelium. And the shape of these are all in cube shape. Basically, it looks like cubes. Okay, so stratified cuboidal epithelium. Transitional epithelium. This is really hard to tell. This is usually occurs in the urinary bladder because the urinary bladder stretches. Transitional epithelium allows the tissue to stretch, so it looks kind of funky. Okay, they look like columnar cells. Let me see if I can blow this up. Make this a bit bigger. Yeah. Okay. It almost looks like columnar cells, but they're not lined up. So transitional epithelium has its own uniqueness to it. So the transitional epithelium allows the bladder to stretch and fill with urine. So it allows it to stretch out as it gets filled up. So that's transitional epithelium. Again, same thing. You can see how all the cells are kind of mismatched. So you can see this is transitional epithelium. And last one. Again, you see the transitional epithelium. Different layers of stuff. All right. Any questions on epithelial tissue? Do you want to go through the quiz with it?
Sure.
Yeah? Okay. Let's see what the quiz is on here. Let's move this over. What structure is highlighted? Like, we haven't done some of these yet, but
Nuclei.
if I can turn the highlights off, I can see them a little bit better, I think. Well, let me turn it off. Oh, there it is. There, I can see it. You gotta learn where everything is on this thing. Okay, so what are those? What's that pointing to?
Nuclei.
The nuclei. Yeah, those are the nuclei of the cells. Okay, perfect. Okay. Which epithelial type is highlighted? I'll turn off the highlights so you can see it. In fact, that's the one I said would be a good test question. What's your first term?
Simple squamous.
Simple. Perfect. Then what?
Cuboidal.
Epithelium.
You got it. Perfect. Simple cuboidal epithelium. It's 1 layer around that lumen. And right here, simple cuboidal epithelium. Okay. Which epithelial type is highlighted? If you look at this, can I make this bigger? No. Area right around here. To me, that's hard to see because it's not big enough.
Mm-hmm.
But it looks like 1 layer, and they look like they're longer than they are, they're taller than they are wide.
Columnar.
It looks like simple columnar epithelium to me. I can't tell real well because it's not big enough. That's what it looks like. Yep, you got it right. I have to blow things up to see them real well. There we go. Which epithelial type is highlighted? Let's turn our highlights on. Okay, so that's the area right there.
Mm-hmm.
So what's that epithelium? How many layers?
Simple.
Perfect. And what shape is it?
Is flat.
Squamous.
Squamous. Simple squamous epithelium. Right here. Yep. You got it. You guys actually did learn something from this. All right. What structures are highlighted? These right here. What are these little things?
They're lagging again. Oh, there it is.
Yep. Those
Nuclei.
Nuclei. Perfect. What structure is highlighted? Turn on highlight. Area right here. So what's the area on the for
Basemental membrane?
basal part. Yep, this would be the basement membrane where the stem cells are located. This is your basement membrane all along here that separates the connective tissue underneath from the cells, from the epithelium. So let's see your basement membrane. What epithelial type is highlighted? Oh, this whole thing. So the first thing I would say would be ciliated, but they don't put that. But it is ciliated. Everybody can see the cilia right here. This is ciliated. So what type is this? Now look at the nuclei. Are they in a straight line?
Pseudostratified?
Perfect, because the nuclei are scattered. So I would say pseudostratified. And what kind of shape are the cells? Are they columns? Are they cubes? Are they squished?
Columns.
Columns.
Columns. So I say pseudostratified. I say ciliated, pseudostratified, columnar epithelium. Since you don't have cilia, I put pseudostratified columnar epithelium. Perfect. Got it. What structure is highlighted? Let's see what the highlights are. You can't tell what it's pointing at. It looks like
Simple Chigro.
it's pointing at the white area, the opening. I don't think it's pointing at the epithelium. I'd say it's pointing at the lumen. And this is through the kidney. This is the kidney section up here. So I would say it's a collecting duct in the kidney. We haven't got that far in class yet, but I think that's where it's pointing to. I think it's pointing to the opening, the lumen. So I would say collecting duct. Incorrect. Basement membrane. I don't see a point in the basement membrane.
I don't see that either.
No, I don't see it. So, like I said, the questions on my test will be non-ambiguous. This is real ambiguous because I don't see the basement membrane. I really don't.
I would think it would be nucleus before do that.
Was that?
Oh, I would say it was a nucleus before the basement membrane.
Yeah, I would too, because it's not actually touching the nucleus. And there's better examples than that too. I mean, it's pointing at something funny. So I thought it was the white area, but oh well. We won't stress over that one. What structure is highlighted? Now I would say what?
Basement membrane.
Yeah, I would say basement membrane.
Basement.
That's the base. That's the portion. There it is. Okay, that is a good example of basement membrane. The other one was not. So if you couldn't see it, don't worry, because I couldn't see it either. But everybody could see this basement membrane, right?
Yes.
Yeah.
This is what it points to, this whole area right here. That's where the epithelial attaches to the connective tissue. All right, what epithelial type is highlighted? All right, perfect. So it's this area right here. And this is going to be...
Screen's lagging. Oh.
Okay. Caught up?
Yes.
Okay, perfect. Okay, how many layers?
One.
Simple.
What's the shape?
Flat.
Perfect. It's flat and squished. It's squamous. So simple, squamous epithelium. Where am I at? Here it is.
Last one.
Simple, squamous epithelium. Yep, got it. Perfect. Hey, what structure is highlighted?
Basement membrane.
Yeah, this whole area right here. I would say basement membrane. Right? There's so many good examples of basement membrane. I don't know why they picked that other one. They had. That's crazy. Because like I said, that your exam questions, it will be obvious. All right, what structures are highlighted?
Nuclei.
Yes.
Yep, those are your nuclei. What? Oh, I hit nuclei. I hit the wrong answer. They're nuclei. I just hit the wrong answer. We said it right. Those are the nucleus of the cell, nuclei. What structures are highlighted here? These are...
It's lightning.
Okay, I'll let it catch up again.
Nuclei again?
Those are nuclei again. Same thing, okay? Perfect. I want to talk fast so this thing can keep up with me. All right, this is one that was really small last time. It's the same section. All right. So how many layers?
One layer.
One layer, because it surrounds this lumen right here. This lumen is full of platelets or whatever cells this is surrounding. So it's the whole area right here. So that's one layer. Okay. And what is the shape of the cells?
Column.
Columnar.
Perfect. Why would this not be pseudostratified? Why is it not pseudostratified columnar? Why is it columnar?
Because they're not next to each other.
Perfect. And the nuclei are next to each other. The nuclei aren't scattered all throughout. The nuclei are lined up pretty symmetrically around it. So I would say simple columnar epithelium. Okay. Everybody sees the difference between columnar and pseudostratified. That's what most students have the most trouble with. Is pseudostratified columnar and columnar. If all the nuclei, which you can see right here, all these nuclei are lined up, which are all pretty much in the line across, then it's columnar. If they're spread out, haphazard across it, some on the apical portion, some on the basal portion, then it's going to be pseudostratified because it looks more like more than one layer. Perfect. And these are what kind of cells? Remember we said
Goblet
secrete goblet cells. Like we haven't gone over those yet, but we will. Which structure is highlighted? Let's check the highlights. Again.
Basement membrane.
I'd say that's the basement membrane. Okay. That's what it looks like it's highlighting. Correct. And I can't blow that up anymore, so I'll do that. Okay. 17. What is the function of a highlighted epithelium? Okay. What is its function? You would have to know more than we have right here. I would tell you where that section is taken from. But this is taken from the epidermis. So if I had to guess, if this looks like skin, it's like that dermal papilla. So if this is the epidermis, this is the outer layer of your skin, what would the function be? Vitamin A, vitamin B12, vitamin D, or vitamin K? Which vitamin? Yeah, that's what I would say. Which vitamin relies on sunlight to hit the skin?
Mm-hmm.
I would say, if I had to make a guess, I would say vitamin D. But we haven't, like I said, and that's correct. Like I said, you haven't. If I asked that of you, I would say this is epithelium from the skin surface. So you can tell this is a dermal ridge right here. This is part of your fingerprint. That's what forms your ridges in your fingerprint. And you don't know that yet. So this is the skin. Skin's responsible for vitamin D production with sunlight. All right, perfect. And now, what is the function of a highlighted structure? First of all, what epithelium is this? We were just talking about that.
Pseudostratified?
Perfect. Pseudostratified columnar. The columnar cells, but the nuclei are all different levels. So it's pseudostratified columnar there. So, but it says what the function of the highlighted material. What's the highlight? Okay, what's being highlighted? What's the structure?
Basement membrane?
Basement membrane.
So the basement, what's the function of the basement membrane? Secretion of mucus? No, secretion of mucus at the surface, right? That's what we secrete and we absorb and secrete at the surface. Contraction to reduce movement? No, those are the microvilli up here, or the cilia, sorry. Absorption of nutrients? No, that occurs up at the top, right? It absorbs through your lumen. There's only one left. Sometimes if you don't that's how you take multiple choice for your nursing exams. You eliminate the ones you know it can't be. If you don't know the answer, just start eliminating them. Resistance of stretching and tearing forces. That's your connective tissue, which is the basement membrane. Okay, everybody see how you get the process of elimination. Even if we didn't know that, we could know these, it can't be. You can't secrete mucus because mucus gets secreted into your lumen or the outer surface. contraction produces movement, that's your cilia. Those all move and they beat, and they produce movement. And so nutrients have to come from the lumen. Absorption and secretion have to come from the cilia. There's only one left it could possibly be. And it's actually right. Okay. Which of the following best describes a highlighted structure? Okay, what's being highlighted?
Basement membrane.
Basement membrane. They like the basement membrane. The basement membrane is being highlighted all across here. So, which of the following best describes the structure? Non-cellular mucus secreting structure. Does it secrete mucus?
No.
No.
No, it's away from the lumen. Okay. Non-cellular supportive sheet. I could go with that. Cellular supportive sheet. I don't see any cells in there, so, and mucus secreting. So it's either cellular or non-cellular. If you take a look at it, look what's highlighted. Do you see any nuclei or any cells in this highlighted section?
No.
No, neither do I. So I would say non-cellular supportive sheet. Oops, wrong thing. Right.
Oh, no.
You see how you can eliminate things when you're taking multiple choice exams, like your N class for nursing school to get in. Eliminate. You know that this structure here cannot produce mucus. This is the lumen. Mucus has to go into the lumen, so you can eliminate those two right there. And if you look at it close, you see there's no nuclei here. If there's no nuclei, there's no cells. So the only one you could possibly put would be non-cellular supportive sheet. Make sense?
Mm-hmm.
Cool. That's what I'm trying to teach you to do, to think, not to memorize stuff. All right. All the cells in the epithelium make contact with what structure? All the cells in the highlighted epithelium. I'm trying to think what that is. I don't recognize it right off the bat. All the cells in the highlighted epithelium. So all the cells in this epithelium. Let me see. I'm trying to highlight it again. Okay, Cook, here's your cell. It's a funny worded question, but we'll figure it out. Okay, so we're talking about all these cells, right? What do they contact? Do they contact the submucosa? Submucosa is way down here below the mucosa.
Basement membrane.
This is your mucosa. This is your submucosa. That's your hypodermis, things like that. So, no. Basement membrane. Do all these cells touch the basement membrane?
Yes.
Yes.
That's a good possibility. The lamina propria is down here. Hyaline cartilage is your cartilage tissue, which I would say basement membrane, since they like basement membrane so much in this course.
Mm-hmm.
Okay, I would say basement membrane, even though I'm not sure, but it's correct. But you look at it and you logically approach your question. You use the knowledge you have to think logically, make a decision. I don't like memorization. When you take my test, they're a lot different than most tests you're going to take. They're not memorization tests. I don't like memorization. I mean, I realize in anatomy, there's a lot of memorization. There has to be. So there is some. Okay? But I like to teach you to think, to logically apply what you know. Because I guarantee you, when you graduate, I don't care what job you get, no one is going to pay you to memorize something. They're not going to give you a book and say, memorize these pages, I'm going to pay you. It doesn't happen. They're going to pay you to think and analyze the business and make them money. They're going to pay you to think, and especially as an RN, you need to think, not just memorize a bunch of structures. But you have to have this as a background.
Mm.
All right, so we finished that. Let's go back to histology and let's go to, we did epithelial tissue. Connective tissue, did we do connective? I think we did some connective tissue already. No, have we done connective tissue?
No.
No.
Okay, we'll kind of go through some of these. We won't do the quiz on this because we don't know all this. We just go through some of these as like an exercise real quickly. Okay, we talked about collagen fibers before. Collagen fibers are your thick fibers running through your mucosa. Okay, so those are your collagen fibers. The real thin ones would be your elastic fibers. Okay, and the fibroblast nucleus, these are your fibroblasts. Fibroblasts make collagen. Okay, fibroblasts produce collagen. Osteoblasts produce bone. Okay. Mast cells, okay, are phagocytic cells. They go through, they're circulating, and they dissolve or eat anything that's foreign in the body. That's what a mast cell looks like. Those are part of your white blood cells. There's 5 different types that we'll learn about. Adipocytes, fat cells. It's going to lag. I can tell it's going to do that. All right, this you should never miss on your nursing exam. Big white cells are fat cells. You don't see a nucleus. You don't see anything. They're just full of fat. Okay, whenever you see this, these are fat cells. They're adipose cells, adipocytes. Okay, they're distinctive. You can't miss it. All right, no one should miss that in your nursing exam. Those are adipose cells, adipocytes. Okay, what are these? I know it's going to lag a second here. These are fat cells again, right?
Mm-hmm.
Big white globular cells. You can see nuclei are squished to the side. There's no nuclei in the center of adipocytes. Okay, they're so full of fat, they squish the nucleus to the side. And you can see the nuclei being squished here, here, here. So all the nuclei are squished to the side. So if you see a section, and nurses love slides like this, okay, in the nursing program, because they're really easy to identify, because they're white cells, sometimes yellow, and there's nuclei squished to the sides. There's no nuclei inside of them. They're just full of fat.
Now, do these replicate or just continue to stretch as they get fuller?
Fat cells, I believe, do replicate.
Oh.
Because you can grow more fat cells. But they will fill up. Yeah, your body can manufacture more fat cells. I'll double check that though. I'm pretty sure it can. Okay, reticular tissue. Okay, you have capsules, trabeculae are your patterns. The big thing with this slide I want you to know is a cortex and medulla. Cortex is always the outside. The cortical layer is your outside layer all the time. This is a lymph node tissue. Your medulla is your inside layer. We talk about the liver. We talk about the suprarenal glands. The cortex is your outer layer. The medulla is the inner layer. We talk about bone. Cortical bone is the outside layer. Medullary bone is the inside layer. It's the same with all our organs. So if we talk about cortex, it means outer layer. We talk about medulla, it means inner layer. Okay, that's the same with any slide we pull up, for any structure. Okay, same thing here. This is your cortex. This is your medulla here. You can see the lymph cells. We haven't talked about those yet. We haven't got into those. Okay, so don't panic on those. There's a lot we haven't gotten to these yet. Okay, these are your lymphocytes. This is what a lymph node looks like. This is when you get your swollen lymph nodes on your neck right here. When you get infections, this is what swells up. These are lymph nodes. Okay, lymphocytes, what's inside of them. Like I said, don't panic here. We haven't done any of this yet, I don't think. You remember, collagen fibers are what? The thicker ones, right? So your collagen fibers, your thicker one. Your fibroblasts make collagen. Your osteoblasts make bone. Okay. So this is skeletal muscle. How do you know this is skeletal muscle? Take a good look at it. We'll do muscle in a little bit here. Why is that skeletal muscle? Not stratified.
Respiration.
But striations. Perfect. You got the ST part right. It's stratified cells. You can see the lines. If I could blow this up, you can see the lines and you're dividing all that. And it's stratified. That's how you know it's skeletal. Connective tissue again. These are fibroblasts. Okay, if we look, I'll get rid of those for a second. We look right here. What is this cell I'm pointing at?
Fat.
Perfect. You got it. Fat cell. Adipose cell. You got your connective tissue, and you got your fibroblasts inside. Again, the fibroblasts are the cells that produce connective collagen. And these are your collagen fibers. You can see how it's all full of fibers here. Let me get off of that. There we go. Okay, dense, some more connective tissue. This is your fibroblast nuclei. Then you see the collagen fibers. Collagen fibers are like waves of fibers that are connected. They're very thick, very long. They produce all the collagen, make it flexible and give the body strength. Add flexibility to the bones. These are your collagen fibers. The fibroblast nuclei, what you'll learn is these nuclei sit. You can see how they all sit in little like depressions. They're all distributed in little depressions. These depressions are called lacunae. I'll go over that in the next chapter. But you can see the fibroblasts all sitting in these little depressions here, little spaces. And again, that's lacunae. Okay. So dense, irregular connective tissue, collagen fibers. Like I said, I'll go through this because we haven't gone over this yet, but we will. Fibroblast nuclei again, collagen fibers. Okay. That's not a good example. I was going to say that, but not a good example there. Okay, this is a cross-section through the aorta. I'm probably going to lag a little bit. Okay, you see a lot of elastic fibers, right? What does that tell you about the function of the aorta? Think what it has to do. What structure receives the highest blood pressure in the body? The aorta, right? That's where the blood comes right out of the heart. The aorta attached to the heart. When that left ventricle contracts, our strongest ventricle, it forces all the blood into the aorta. And from there, it gets distributed to the cranium, to the stomach, to the rest of the body. And the blood pressure drops. The greatest amount of force you're ever going to have is the aorta. It has to stretch. Perfect. Okay? It has to stretch. It has to be able to handle that force. Anybody hear of an aortic aneurysm? Aortic aneurysms are usually fatal. You bleed out instantly. I mean, you have minutes to live, if that long. Because if this ruptures, all the blood that's going to your body comes out of it. It has to stretch. That's why if you look at the histological slide, you see all kinds of elastic fibers. So if you saw that on a test, you can make a good guess that that's probably an aorta or something similar, one of the large blood vessels. It would not be a vein. Veins would not have blood. They have to stretch a little, but not as much that aorta. So that you can see all these elastic fibers that run through there, all these screw things, that allows it to stretch. And it handles that blood pressure that goes through from the left ventricle contraction. Okay, this is a different stain. Okay, this is an iron hematoxin stain, so it's a darker stain. But again, you can see all the elastic fibers. The other was an H&E stain. This is a hematoxin iron stain. So it's the same thing. You see all these fibers here. Those are all elastic fibers because it has to be able to withstand a tremendous amount of force when that heart beats. You don't want the aorta to rupture. Again, the elastic fiber is kind of more blown up. You can see all like squiggly structures in here. And they just allow this organ or this structure to expand. And we'll go over the heart and go over all that. We do the heart. I think we have 2 or 3 sections on the heart. Hyaline cartilage. Okay. Hyaline cartilage has a perichondrium. A perichondrium is the outer layer surrounding the structure. Okay, so that's the perichondrium is the outer structure of hyaline surrounding it. And this is hyaline cartilage. This is the cross section of your trachea. Okay, you see all these little ciliated cells here? That's what causes the mucus to be produced. And all they beat is they beat in one direction and force that mucus out of your lungs. Any materials, any pathogens, anything like that. The hyaline cartilage is what surrounds the trachea. Remember we said they were cartilaginous rings. The trachea never collapses. Okay, the esophagus are always collapsed. When we put food in it, it forces it open. The trachea never collapses. It's an open tube. Okay, air can flow freely through it, cannot collapse. This is why it can't collapse right here. These are cartilaginous rings that are spaced along the trachea. Cartilage is very hard. It's not as hard as bone, but it's still hard. Okay, and it prevents the trachea from collapsing. So it keeps it open. When we talk about a cricoid membrane puncture as a nursing student or as an RN, you may have to do one. And you feel the Adam's apple and you feel the space between the cartilage, between those cartilages. You have to go between the cartilage. You can't cut through the cartilage. You feel that space, and that space is right there. You just stick right in between there. Right under your Adam's apple, you'll feel cartilage above and cartilage below. This is called the cricoid membrane. It's a membrane between the cartilage. Okay? You're not going to penetrate this cartilage. Look how thick that stuff is. Okay? You got to go between the cartilaginous rings. It's called a cricoid membrane puncture. Okay? And it bypasses the tracheal rings. Okay. Remember, I mentioned lacunae. Okay. All the fibroblasts, what produce cartilage, live in or reside in little depressions in the cartilage. And those are called lacunae, Lacunae. Okay. And it's the exact same thing for osteoblasts. They make bone. Lacunae also occur in bone. So that's one of the similarities, on your nursing exam again, one of the similarities between cartilage and bone is they both have lacunae. That's one of your similarities. Osteoblasts live in lacunae, fibroblasts live in lacunae and cartilage. Okay? And you can see how thick the hyaline cartilage is. And the pericardium is the outer layer of epithelium surrounding the hyaline cartilage. Okay? But you can look at all these little areas in here, and you can see all those little depressions inside, and it secretes a matrix in between here. We see all the cells live in these little spaces called lacunae.